Hello and good afternoon friends, welcome to CEC EduSec live lecture. Dear friends, as you know that this session of ours is all dedicated to the science subjects, and as you also know that we are talking lots on botany, that is we are catering to the botany subject, and today we are going to discuss on plant physiology, and under this broad topic we are going to discuss on phloem translocation. We are going to know what basically phloem translocation is, and for this we have with us in our studios Dr. Madhumita Banerjee. She is Associate Professor in Ramjas College, Department of Botany, Delhi University. So first of all, I would like to welcome our guest Dr. Madhumita Banerjee to this EduSec lecture of ours. Hello ma'am, welcome to the EduSec lecture, and as we are going to discuss on this phloem translocation, and as per the discussion with you that you explained how the sugar produced during the photosynthesis process, and then it is we can say that located in the different parts of the plant. So we are going to know through you how this process works and what are the benefits of it. So ma'am, over to you. We would be like to hearing from you.
Thank you, Geetika. So the topic for discussion today is phloem translocation. Now life evolved in water, and plants, when they migrated to land, they faced many challenges in their terrestrial existence. To meet these challenges, plants developed roots for anchorage and absorption of water, and leaves for photosynthesis and exchange of gases. Now, as the distance between the roots and leaves increased, they developed specialized tissues. They developed xylem tissues for long distance transport of the water that is absorbed by the roots, and they developed phloem tissues for long distance transport of the photosynthetes manufactured in the leaves to be transported to all parts of the plant. So phloem translocation is basically the transport of photoassimilates over long distances, and this takes place in the phloem, and the translocation takes place from a source to a sink cell. Now, in this course of this discussion, we will focus on different aspects of phloem translocation. We will discuss the experiments that first proved that translocation does take place in the phloem. We will study the anatomy of the phloem, and we will study the nature of the translocated material and the rates of transport. And then we will try to understand the source and sink concept, and next we will go on to the translocation process itself, which takes place in 3 steps. First one is phloem loading, which takes place at the source. The second one is the long distance transport through the phloem, which is basically transport from the source to the sink cells, and we will try to understand the bulk flow hypothesis, which gives an explanation for this process. And the third step in translocation we will also discuss, and that is phloem unloading, which takes place at the sink cells, and we will conclude with a discussion on allocation and partitioning of the photosynthetes. Now, the earliest experiments on phloem translocation was one the girdling experiment. In this experiment, a ring of bark containing the phloem was removed from the trunk of a tree, leaving the wood intact. Now, as a result, the transport of water absorbed by the roots from the roots to the leaves was not affected, but the translocation of photosynthetic material from the leaves to the roots was interrupted, and the photosynthetic material it accumulated on the region just above the girdle, and that portion became swollen as a result. So this experiment for the first time indicated that translocation actually takes place in the phloem tissues. Then experiments with aphids also helped scientists to understand the nature of the translocated material. Aphids are small insects. They can be xylem feeders or they can be phloem feeders. Now, the phloem feeders, they insert their mouth parts, which consists of 4 stylets, into the phloem, and they suck the phloem sap. They feed on the phloem sap. Now, this stylet was cut while the insect was still feeding, and the phloem sap, because the sap in the phloem is under pressure, the phloem sap oozed out of the stylet, and the sap was collected and analyzed. It was found that that sap was rich in sugars, again proving that sugars are translocated in the phloem, and this experiment also proved that the phloem sap is under pressure. A third experiment used radioisotopes. Leaves were allowed to photosynthesize in the presence of 14 CO2, and the sugar for 10 minutes, the sugars became labeled, and they were localized, the radioactivity was localized in the phloem tissues, and this conclusively proved that translocation takes place in the phloem tissues. Now, in order to understand this process of phloem translocation, we must understand the anatomy of the phloem tissues. Now, the phloem is a complex tissue, and in angiosperms, the phloem is made up of the sieve tube elements, the companion cells, phloem parenchyma, and in some species there are also phloem fibers. The sieve tube element is unique because it is a living cell, but at maturity it does not have a nucleus. Now, the sieve tube elements, a number of sieve tube elements are joined end to end to form the sieve tube, and in between 2 adjacent sieve tubes we have the sieve plate, and in the sieve plate there are pores. There are pores in the sieve plate. These pores are open, and therefore these pores provide a continuous channel for the translocation of photosynthates through the phloem from the source to the sink. Now, in the sieve element there is also a protein, a special type of protein which is only found in the sieve element, and it is called the P protein. This protein could be tubular, it could be fibrillar, but this P protein in an intact sieve tube element, it is located along the inner walls, and they do not block the pores remain as open channels. But when the sieve tube element is injured, then the P proteins may block the plugs, and that actually helps in reducing the loss of the phloem sap. So, in addition to these P proteins, sieve elements also have callose, and the callose also plays an important role in sealing off damaged sieve tube elements to reduce loss of phloem sap. Now, we must understand the structure of the companion cells. The companion cells are very closely associated with the sieve tubes, and the structure of the companion cells is important because it determines the type of phloem loading that will take place. Now, these companion cells, they have a number of plasmodesmatal connections with the adjacent sieve tube element, and there are 3 types of companion cells, and all of these 3 types of companion cells are symplastically connected, that is, they have plasmodesmata connections with the sieve tube element. However, the 3 types of companion cells, the first type is the ordinary type of companion cells, And this type of companion cell, while it is connected to the sieve element, it does not have any connection with the surrounding mesophyll cells and the bundle sheath cells. It does not have any plasmodesmatal connection with them, and therefore we say that the ordinary type of companion cell is symplastically isolated. The second type of companion cell is the transfer cell, transfer type of companion cell, and it is very much like the ordinary companion cells, except that they have finger-like outgrowths on the walls, on their walls. But here also, in the transfer cells also, they do not have connections, they do not have plasmodesmata connections with the surrounding cells, that is, they are also symplastically isolated from the surrounding cells. Now, the third type of intermediary of companion cells are the intermediary companion cells. And these intermediary companion cells, they are of course connected to the sieve tube element, but in addition they have many plasmodesmata connections with the surrounding cells, especially with the bundle sheath cells. Therefore, this type of companion cell is not symplastically isolated, and it is found in plants where phloem loading occurs through the symplast. Analysis of the phloem sap gives a fair idea of the material that is translocated in the phloem, and such analysis has shown that it is the sugars that are in highest concentration in the phloem sap, and these sugars are all non-reducing sugars like sucrose, raffinose, stachyose and verbascose. Reducing sugars like glucose and fructose are not translocated in the phloem. This is probably because these reducing sugars are more reactive and can be easily broken down by the enzymes present within the phloem. Other than these non-reducing sugars, sugar alcohols like inositol and mannitol are also translocated in the phloem, and amino acids and amides like glutamine and glutamic acid are also translocated, and in some plants ureides are also translocated within the phloem. And in addition to this, many minerals like potassium, chloride and magnesium are translocated to the phloem, and hormones, the naturally occurring, that is the endogenous hormones, are translocated. Cytokinins, auxins, gibberellic acid, abscisic acid are all translocated through the phloem. Now, the rate of phloem translocation is important. It is to know this rate is important because it is quite high. It is 1 meter per hour on an average, and this rate is much faster than the rate of diffusion, and therefore this rate indicates that phloem translocation does not occur by simple diffusion. Now, what is the direction of phloem translocation? Phloem translocation always takes place from a source to a sink. So, what is a source? A source is an area of supply. It is any organ that produces, assimilates in excess of its own needs. All right. So, a source could be actively photosynthesizing leaf, could be a source. Now, tubers, when they are sprouting, they are export organs, and when they are sprouting, a tuber is a source. Similarly, a germinating seed is also a source. So, what is a sink? A sink is a non-photosynthetic organ that imports sugars which it requires for its own metabolic activities or for storage purposes. For example, the root is a sink, And even organs like seeds, when they are developing organs like seeds or tubers are also sinks. A sink can become a source at a certain point of time. For example, a leaf, when it is very young, it is a sink, but when it is older and it is actively photosynthesizing, it becomes a source. So, to conclude, we can say phloem translocation always takes place from a source to a sink, and the direction of translocation could be either upward or downward, and it is this direction of translocation is not directed by gravity. Now, we come to the translocation process itself, and this transport of the assimilates from the source to the sink takes place in 3 steps. The first step, as we have already said, is phloem loading, and this is a short distance transport which takes place at the source. Then is the second step is the translocation from the source to the sink. This is a long distance transport, and the third step is phloem unloading, and this too is a short distance transport, and this takes place at the sink. Phloem loading. What is phloem loading? The phloem loading is the process by which sugars are loaded from the source cells into the sieve tube elements, and this can take place via 2 routes. One is the symplastic route, which takes place through the plasmodesmata connections, and the second is the apoplastic route, where the sugars pass from the source cell to the cell wall and then enter into the sieve element. So, as we can see in this slide, that photosynthesis produces, due to photosynthesis, sugars are produced in the source cells, and they move from one source cell to the other through the plasmodesmata connections, and then when they enter the companion cell through the plasmodesmata connections itself, that is symplastic phloem loading. However, in certain cases, the companion cell does not have a plasmodesmata connection with the surrounding cells. In that case, the sugars enters into the apoplast, that is into the cell wall of the companion cell, from where it is loaded onto the sieve element, and this is called apoplastic phloem loading. So, we can just conclude by saying that apoplastic phloem loading takes place in those species where there are ordinary companion cells or transfer cells, and here apoplastic phloem loading takes place because these companion cells do not have any plasmodesmata connections with the surrounding cells, and in such species showing apoplastic loading, sucrose is the main sugar that is translocated. In plants which have symplastic loading, they have the intermediary type of companion cells, and in such plants their sugars that are translocated are sucrose, could be sucrose, but in addition to this other oligosaccharides are also translocated through the intermediary type of companion cells. So, for phloem loading, for symplastic, to explain symplastic phloem loading, a model has been suggested, and that is called the polymer trapping model. According to this polymer trapping model, there is a high concentration of sucrose in the bundle sheath cells, and this sucrose enters the intermediary cells through the plasmodesmata connections. In the intermediary cells, the sucrose is used to synthesize raffinose or the oligosaccharide stachyose. Now, This, because the sucrose is utilized in the synthesis, the gradient of sucrose between the bundle sheath and the intermediary cell is maintained, and the oligosaccharides raffinose and stachyose, they cannot flow back into the bundle sheath cells because of their larger size. The plasmodesmatar connections between the bundle sheath cells and the intermediary cells do not allow the larger oligosaccharide to go back to the bundle sheath cells, and the raffinose and the stachyose enter into the sieve element through the plasmodesmatal connections between the intermediary cell and the sieve element. This model for symplastic loading, the polymer trapping model, has been validated because it has been seen that many predictions made by this model is true, and it has been found that as predicted by this model, sucrose is more concentrated in the mesophyll cells than in the intermediary cells. Secondly, the intermediary cells have enzymes for the synthesis of raffinose and stachyose. And thirdly, the plasmodesmata connecting the bundle sheath cells and intermediary cells exclude molecules larger than sucrose. So, the polymer trapping model has been validated, and it is the process, it is the accepted way in which symplastic phloem loading takes place. Apoplastic phloem loading, in this process, in the apoplastic phloem loading, sugars enter the apoplast at some point during their transport from the source cell to the sieve element companion cell complex. As shown in this slide, in apoplastic phloem loading, the sugars exit the source cell and enter into the cell wall of the companion cell, and from the cell wall of the companion cell they enter into the phloem, and this process is an active process, and H+ sucrose symporters are present which help in transporting the sucrose from the apoplast into the companion cells. So, this apoplastic phloem loading has also been validated because its predictions have been validated, and therefore it has been accepted. And the predictions that it makes is that it has been found that transported sugars are present in the apoplast, and sugars which are applied to the apoplast do actually accumulate in the sieve elements and companion cells, and inhibition of sugar uptake from the apoplast results in inhibition of sugar export from the leaf. And symporters, this apoplastic phloem loading model has also been accepted because sucrose H+ symporters have been isolated from the plasma membrane of the companion cells in the case of Arabidopsis, and from the sieve elements in tobacco, tomato, and Arabidopsis also. Now we come to the second step in the translocation process, that is the movement, the long distance movement of the sugars from the source to the sink, and this process can actually be observed in a living sieve element. This is done by making two windows on the vein, and in the apical window one inserts a phloem mobile dye, and then the translocation is observed through the basal window, through the second window, by inserting through a confocal microscope. So, in this way translocation can actually be observed. Now, to explain this long distance translocation, Munk proposed a theory, and this theory was based on a model. The model given by Munk consisted of two osmometers which were connected to each other, and one of the osmometers had a higher concentration. Both the osmometers were immersed in pure water. One of the osmometers had a higher concentration of sugars, and the other osmometer had a dilute sucrose solution. Now, it was observed that the flow of water into the concentrated sugar solution exerts a pressure, and this pressure causes the concentrated sugar solution to flow to the other compartment. So, based on this model, Munk proposed the bulk flow hypothesis to explain phloem translocation, and he said the same situation exists in the plant, and the osmometer with the concentrated sucrose solution is equivalent to the source, and the osmometer with the dilute sucrose solution is equivalent to the sink cells. So, the phloem translocation is explained by Munk, and he has given the bulk flow hypothesis or the pressure flow hypothesis to explain this. And according to this hypothesis, the translocation is driven by an osmotically generated pressure gradient from the source to the sink. The pressure gradient is established due to phloem loading at the source and unloading at the sink, and this is a passive process. It does not require any energy. So, if we look at the phloem tissues, as this diagram shows, that due to phloem loading at the source, the concentration of sugars in the sieve elements at the source becomes very high. Consequently, the water potential becomes very negative, it becomes very low, and as a result water enters into the sieve elements at the source. As a result, the sieve elements at the source develop a very high turgor pressure. On the other hand, in the sink, what is happening is that the phloem unloading is taking place. The sugars are transported from the sieve element into the sink cells. As a result, the water potential of the sieve element at the sink end becomes more positive, and when the water potential becomes higher than the water potential in the xylem, the water leaves the phloem and enters into the xylem. Now, when the water leaves, the turgor pressure in the sieve element at the sink end becomes low. So, as a result of this, there is a pressure gradient, a pressure difference between this in the sieve element at the source and at the sink. There is a higher pressure at the source end and a lower pressure at the sink end, and the gradient has been found to be in the range of 0.1 megapascals to 0.4 megapascals, and it is this pressure that forces the translocate the sugars or the contents of the phloem that actually physically forces the contents of the phloem from the source to the sink end, and the sugars just move by mass, by bulk flow. They are transported from the source to the sink. Now, if we closely inspect the water potential values, we will see that the water in the phloem is moving against a water potential gradient from source to sink. But this does not violate any the laws of thermodynamics, as water flow in the sieve element is driven by a pressure gradient, and it is rather than a water potential gradient. So, as per the pressure flow hypothesis, the phloem translocation is a passive process. Now, there are certain predictions that have been made by the pressure flow model, And for the pressure flow model to be accepted, the sieve plate pores must be unobstructed, because if the P proteins or other materials block the pores, then the resistance to the translocation would be great, and the translocation could not take place. But the pressure flow hypothesis has been validated, because it has been shown that the C pores are not blocked by P proteins, but they are open channels. Second prediction made by the pressure flow hypothesis is that bidirectional flow in a single sieve tube cannot occur. This too has been validated, and it has been found that bidirectional transport in a single sieve element does not ever occur. However, transport or translocation can occur in two directions, and this would take place in sieve elements of different vascular bundles. The third prediction is that the process should not be energy dependent, and this too has been validated, because when metabolic inhibitors like cyanide is used, which stops ATP production, this does not have an inhibitory effect on phloem translocation. And fourth, this model predicts that there should be a positive pressure gradient from the source to the sink. This too has been validated, and it has been found that the pressure gradient that is required is between 0.12 to 0.4 megapascals, and that actually exists in the it is found to be actually present, that pressure gradient is present. And therefore, because the pressure flow hypothesis is accepted, and this is the explanation for translocation of the solutes from the source to the sink cells. The last step in phloem translocation is phloem unloading. So, phloem unloading is basically the process by which the sugars will exit from the sieve element and will enter into the sink cells. Again, this can take place in two ways. One, there can be symplastic unloading, or two, there can be apoplastic phloem unloading. In symplastic unloading, the sugars will exit from the companion cell sieve element complex via plasmodesmatar connections into, and they will enter into the sink cells and then pass from one sink cell to the other through plasmodesmatal connections, and that is why this is known as symplastic phloem unloading. Apoplastic phloem unloading also takes place, and in apoplastic phloem unloading can be again of two types. Here in the first type, the transport from the sieve element companion cell complex to the successive sink cells occurs in the apoplast, and once the sugars are taken back into the symplast of adjoining cells, transport is symplastic. So, in the first process, first type of apoplastic phloem loading, the sugars exit the sieve cells and enter the sink cell through the apoplast, but further transport from one sink cell to the other is through the symplast. In the second type of apoplastic phloem unloading, sugars exit the phloem and enter the sink cells through the plasmodesmatal connections, that is, it is symplastic. But at a later stage, when the sugars move from one sink cell to the other, there is an apoplastic step which may be located close to the phloem or maybe a little far away from the phloem. So, the photo assimilates Now this translocation process is complete. The photoassimilates have been translocated from the source cells to the sink cells. Now these photoassimilates are distributed between different metabolic pathways or between different plant organs, and the distribution between different metabolic pathways is known as allocation, while the distribution of the photosynthate between different plant organs is known as partitioning. Now, so allocation can take place both at the source and or in the sink cells, and these allocations may be for metabolic utilization. For example, the sugars can be used for respiration, or the allocation may be for synthesis of storage compounds, that is short term storage, or the sugars can be allocated in a source cell. They can also be allocated for export to other parts of the plant. So allocation can take place both in source cells and also in sink cells. Now, regulation of allocation is a very complex process, and it involves the interaction of a number of pathways, and it is also within a source cell, it is also genetically programmed. Now, distribution of the photosynthetes between different organs, as we have said, is referred to as partitioning, and the sinks, different sinks, they compete for the photosynthate, they compete to get a part of the photosynthate material, and sinks which have a higher strength, they get more of the assimilates than weaker sinks. Now, so what is a strong sink? A strong sink, sink strength is given by the sink size and sink activity. So, the larger the sink and the more active the sink is, that is, the more actively it is using up the sugars, it is a stronger sink. A stronger sink increases the rate of translocation towards itself, and translocation from a particular source to a particular sink depends on the position of the sink in relation to the source. So, a sink is always connected to the nearest source. For example, if the sink is a root and the source is the leaf which is towards the base of the plant would be supplying the root as a sink. And another factor that determines source sink linkages is vascular connections. A source will connect to that sink with which it has a good vascular connection. So, phloem loading and unloading and photosynthate allocation and partitioning, these are of great research interest because they play a very important role in crop productivity. And I would just like to mention that I have taken, my references are from Hopkins and Huner, and also from Tez and Zeiger.
Thank you, ma'am. Thank you so very much for delivering such a nice lecture, and you explained how basically the translocation takes place. But if in simple terms we talk about translocation, if we think about our students are unknown with this particular term translocation, so how would we describe or define this particular term?
Translocation in this context, in the context of phloem translocation,
exactly,
simply means how sugars are transported from a source cell to a sink cell through the phloem tissues.
And this process takes place at what particular time?
This process can, when the sugars are manufactured during the day, photosynthesis takes place, and it is during the night that the translocation will take place, and the sugars are converted into mobile forms and sucrose is synthesized and it is then transported.
Once this process is done, what is the further step? What are the benefits of this sugar translocation if we talk about within the plants?
Because see, only the leaves are capable of manufacturing the sugars, and all other organs are dependent on that sugar for all their requirements, for their metabolic requirement and for their growth, and even if they want to store it. So for all these requirements, they are dependent on the sugars that are manufactured in the leaves. So translocation is a very important process.
If you see, said so, the translocation is an important process. Sometimes we have seen that a plant which tends to grow at a more height, but it does not. We can say that the translocation process would not have been taken properly. That's why the growth of the plant would not have taken properly.
Yes, if there is no translocation, growth will be affected. Of course, growth will be affected, and we can say that. And in fact, if as you see on the girdling experiments, if you remove the phloem and if actually you stop translocation, then the plant will die. Ultimately, that part which is not getting the sugars will not survive.
Some fruit bearing plants, if we talk about, does this translocation of sugar does further improves the quality of the fruits?
Yes. In fact, a lot of agricultural scientists, they try to do research on trying to improve agricultural products. So if you can have more translocates or more sugars coming into the fruits, you will get a better quality of fruit, and there is research going on this aspect.
It is the process which we are talking about. It is the natural process
it is
with which. But would it be possible if for the special plants which grow under the special conditions, if we talk about especially the plants which grows in the polyhouses, now there this process there is any difference?
No, the process is basically the same. The process is the same in all plants. This is how it takes place, and it is a natural process. But when a translocation takes, with a source, as I told you, a source can become a sink also, and so you can sort of enhance the process and get a better product. That you can do. But the process is the same in all plants. Yeah.
So ma'am, what else more we can discuss in the plant's physiology, as we discussed on the plant, this phloem translocation, what could be we can learn through you more in the future lectures?
On plant translocation?
On plant translocation or the plant physiology.
We can talk about photosynthesis, how photosynthesis is, that's the first thing that happens, and then the sugars are manufactured, and this is the, this comes afterwards.
This comes afterwards.
Definitely,
ma'am, in future lectures we would be discussing on photosynthesis and all the processes which take place after the phloem translocation. And with this note, I would like to thank you so very much for being with us and giving us deep insight into this very topic. And I hope that all the students who might have watched this particular lecture and would be watching us through the YouTube would be benefiting from this lecture. Thank you, ma'am. Thank you so very much.
Thank you, Geetika.